Hi folks, welcome to the last installment of A Tale Made of Biomolecules, How Structure Determines Function. And today we're going to be talking briefly about nucleic acid. I want to foreshadow this by saying that we're going to be exploring nucleic acid structure in more detail when we talk about how DNA replicates and how genes are expressed, transcription and translation. But this serves as an introduction. So nucleic acids are polymers of nucleotides. On the right, you will see a picture of one such nucleotide right here. On the left, hopefully you will recognize this as part of a DNA molecule. So what does a nucleotide look like? And this is the most famous nucleotide called ATP, adenosine triphosphate. It is composed of a nitrogen-containing base, in this case it's adenine, which is linked to a 5-carbon sugar, in this case a ribose. The ribose is now linked to 3 phosphate groups. The presence of phosphate groups in nucleic acid is what gives them the name acid, because remember, phosphate groups are negatively charged and acidic. They donate their protons, they donate their hydrogen ions to the solution, thereby raising the hydrogen ion concentration. And here's the naming convention. If you just have the base and the ribose, the sugar, you're called adenosine. If you have the base, the sugar, and a single phosphate a single phosphate group, you're an adenosine monophosphate. If you have 2 phosphate groups, you're an adenosine diphosphate or ADP. If you have 3, you're an ATP, adenosine triphosphate. Now, ATP, you might have learned, is the molecule of, that cells use to store and obtain energy, the energy currency. However, ATP is a nucleotide, and therefore ATP can be used to build nucleic acid polymers. And because ATP contains ribose, hopefully you'll know that that type of polymer is called RNA. So what's the difference between DNA and RNA? You know the major differences between DNA. DNA is double-stranded, RNA is single-stranded, DNA has T, RNA has U. You've learned all this in bio, but I would like to focus on that one particular difference, the difference in their sugars. DNA has deoxyribose, and that's where the name comes from, deoxyribonucleic acid. RNA has ribose, that's why it's called ribonucleic acid. So what is the difference between these sugars? A ribose or deoxyribose are pentoses. They're 5-carbon sugars. Remember, sugars in solution form rings. Rings have their carbons numbered. And I want to remind you that we started the oxygen here at the top of the ring, and we're going to go clockwise. This is the 1st carbon, the 2nd, the 3rd, the 4th, and the 5th is sticking out. Same thing here. 1st carbon, 2nd carbon, 3rd carbon, 4th carbon, the 5th one is sticking out here. Again, we started oxygen and go clockwise. So what do you notice? At the 2nd carbon, where it says 2', deoxyribose has a hydrogen. Ribose has a hydroxyl group. So the presence or absence of a single functional group, remember, can make or break someone, right? It changes the property of that molecule, and therefore it changes its function. So what properties have we changed here? Because ribose has a hydroxyl group in this position, it is subject to hydrolysis, so breakdown by water. So ribose containing molecules are less stable. Deoxyribose does not contain a hydroxyl group in this position, and therefore is more stable. So the 2 sugars vary only slightly in structure, but the properties are very different. And because their properties are very different, their chemical properties are different, their functions are different. Ribose is used in RNA, deoxyribose is used in DNA. So why are we using deoxyribose in DNA? Or rather, the question is, why are we using DNA to store and carry our genetic information? Because the answer is Deoxyribose is more stable than ribose. So DNA is the preferred method to store and transmit genetic information because it's more stable molecule than an RNA molecule. And why? Because of the presence or absence of a single functional group on the sugar of the nucleotide. So again, this is a great example how structure determines function. Finally, a little bit of a trivia. Why do we call this deoxyribose? Because it literally means deoxy means without oxygen. So here we don't have oxygen, here we have oxygen. However, I didn't want to start with this because you know better than this. It's not the presence or absence of oxygen, it's just that the functional group here is hydroxyl group. Here, we do not have a functional group, we have a single atom. And it is the presence of this functional group that changes the property of this particular sugar, makes the properties of this particular sugar different than the properties of this sugar, and therefore changes the function of molecules that contain either sugar. So the components of a nucleotide reviewed: a phosphate group, a sugar, again, in DNA it's a deoxyribose, and RNA is a ribose, and any one of 5 bases, or rather 4 bases, because in DNA it's thymine and RNA is uracil. So thymine is substitute for uracil in RNA. The bases are 2 classes. Purines are bigger ones. So the word purine is shorter than pyrimidine and easier to pronounce, but the bases are bigger. They contain 2 rings, so they are larger. The pyrimidines are a single nitrogen-containing ring, so they're much smaller than the purines. And this matters. We'll see soon enough why that matters, the size of the bases. So a nucleotide is made of a base, a sugar, and a phosphate group. The polymers of DNA and RNA are really linear polymers of nucleotides, and they're linked by a 3', 5' phosphodiester bond. And I'm going to explain what that is in more detail when we talk about DNA synthesis. Right now, all you need to know is that basically a phosphate group will link 2 adjacent nucleotides. The polynucleotide, which is formed by this process, has directionality. Just like proteins, head to tail, nucleotides are also linked head to tail. So one chain will start with a 5' end and end with a 3' end. The 5' is the 5th carbon where the phosphate group is attached. The 3' is the hydroxyl group of the 3rd carbon in the sugar. And I will show you again. 1st carbon, 2nd carbon, 3rd carbon, 4th carbon, 5th carbon. So on one end of the molecule, you're going to have the phosphate group, which is attached to the 5th carbon. So it's called 5' end. On the other end of the molecule, you're going to have a hydroxyl group, which is free. It is not attached to anything. It's only linked to the 3rd carbon, so this is the 3' end. The reason we're talking about directionality is because this phosphate group is going to attach to this hydroxyl group of the next nucleotide. So the structures of DNA and RNA molecules differ in more than just the sugar. RNA molecules usually exist as single chains, whereas DNA molecules have 2 chains spiraling around an imaginary axis and forming a double helix. The DNA double helix has 2 backbones, and the 2 backbones run in opposite directions, and this is very important. One runs in 5 to 3, the other one will run in 3 to 5. This arrangement is referred to as anti-parallel. One DNA molecule can include many, many different genes. And here is a picture of such a DNA molecule. What is in purple is called the sugar phosphate backbone. If you look at the first row, you will see 2 nucleotides across from each other. The base, the sugar, the phosphate group. The base is sticking out. The base on this nucleotide is also sticking out. The base on this nucleotide is also sticking out. What really forms the polymer is the phosphate group and the sugar. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. It's the phospho-sugar backbone. And it runs in the 5 to 3' direction. Why? Because the first nucleotide will use its hydroxyl group on the 3rd position, right here, the 3rd carbon, and form a link with the phosphate group of the next nucleotide. This nucleotide will use its hydroxyl group and form a link with the phosphate group of the next nucleotide, and so on until you reach the very end. The very last nucleotide will have used its phosphate group to form a covalent bond with the previous nucleotide, and its hydroxyl group, okay, let me just wait a second. Okay, the hydroxyl group of the last nucleotide, which is attached to the 3' carbon, the 3rd carbon, remains free. It's unattached. So this will be the 3' end of the molecule. The other DNA chain is really flipped. It goes from 3 to 5. And the reason that is, we're going to explore more. But what I want you to notice is that the 2 chains are held together by interactions between the bases, and the bases are on the inside. The bases form hydrogen bonds between polar groups in the base. And there's 2 hydrogen bonds between A and T, and 3 between C and G. And an A always pairs with a T, and a C always pairs with a G. And what I also want you to notice that the A is a big base, it has 2 rings, and it pairs with a single ring, a smaller base. Similarly, C is a small base, a single ring, pairs with G, which is a bigger base, it has 2 rings. So in each case, you have a smaller base paired with a bigger one. So what I just explained was really the base pairing rule. So the base pairing rule, DNA pairs out via hydrogen bonds between A and T, 2 of them, and between G and C, 3 of them. And this is called complementary base pairing. This is all a review for you guys because you know this from bio. However, what you may be not knowing, not have thought about or not know is that complementary pairing between bases can also occur between RNA and DNA, between RNA and RNA, or between parts of the same molecule. So an RNA molecule can fold on itself and base pair, and so can DNA. There's nothing to prevent that. Also reminding you that the T, the thymine in RNA is really replaced with an U, so the base pairing in RNA is A to U, not A to T. Okay, and here is the picture again. I really want to stress how important it is that you know that the 2 polynucleotides in a double helix DNA run in opposite directions. This is going to become very, very important later. Okay, so here in the upper picture is an example of base pairing. So here we see base pairing between 2 DNA strands. Here on the upper picture, you will see base pairing between an RNA and a DNA strand. So as RNA polymerase transcribes a gene, so it basically takes the information written in DNA and transfers it to an RNA molecule. It does so using the base pairing rule, using complementary bases. And there is an RNA-DNA hybrid, so it's basically a double-stranded molecule composed of 1 strand of DNA and 1 strand of RNA, temporarily formed so that RNA molecule can be made. So the double helix of DNA is melted, and now one of the strands of DNA is used as a template to synthesize an RNA molecule. And that occurs via base pairing, complementary base pairing. Complementary base pairing can occur within the same molecule in DNA as well as in RNA. So in a cell, we always say RNA is single-stranded, DNA is double-stranded. In reality, although RNA is single-stranded, RNA is never fully stretched out. RNA forms complex structure by folding on itself using base pair complementarity. And in this cartoon, what you will see here is that one part of the RNA molecule base pairs with a different part of the RNA molecule, forming a kind of like a structure. And this is important because the structure of RNA also determines its function. We're going to see this when we talk about how messenger RNA molecules are translated by the ribosome to make proteins. Okay, and I'm going to end this here by reminding you that, yes, DNA is where all the information is stored for how to build proteins. RNA molecules carry that information to the cytoplasm. RNA molecules build ribosomes. RNA molecules help translate that information and make proteins. So RNA molecules are extremely important. Let's not discount them. But this is the end for our video tutorial on nucleic acid structure and function. More to come when we talk about DNA replication and gene expression. For now, goodbye.